pandemic has hit the United States and the entire world. It kind of took us by surprise, but many people, many scientists had predicted that something like this may have been coming, it may have been in the works, but I guess we weren't, as a species, ready to deal with this, and the effects are really difficult. This virus is very resilient. Many studies have already been published over the last 2, 3, 4 months, demonstrating that the coronavirus is very stable for several hours to several days when it's either airborne or stuck on specific surfaces. As we here in the US and the world over are trying to really reopen up our business, create a much healthier environment to allow people to go back to work, to allow life to return to the new normal, hopefully somewhat representing the old normal. We understand the need of testing. We have to be able to identify exactly where the virus is, exactly how it behaves, exactly how it transmits. And to do that, we have to have accurate, sensitive, and timely testing. That's the only way to help businesses, organizations, governmental agencies to be able to return to work and resume the normal duties they have had in the past. In order to be able to do this, we have to understand more about this coronavirus. And for that, I'm going to turn it over to my colleague, Dr. Hari Bezari, to tell us a lot more about COVID-19. Hari?
Well, thank you, Paul. Thank you, Dennis, and thank you everybody for joining us. The last couple of months, every time we turn on the TV, we hear the word testing for COVID-19 virus. And what is this testing? The testing is a very broad word, actually. So hopefully in this webinar today, we're going to put some light on these various testing methods that has been utilized to test for the COVID-19. But what are we testing? We're testing for the coronavirus. And so the first thing we need to know is what is virus. Virus is an infection agent, is very small, it has a very simple composition, and can only multiply on the living cell or living organism, which is called the host. The host could be an animal, it could be a plant, it could be a bacteria, it could be a human. Virus particles are made up of genetic material that are housed inside the protein shell. And this genetic material actually are called genome. Virus may consist of the single strand or double strand DNA or RNA. They might be maybe linear, they could be circular. And now talking about DNA and RNA, what is DNA? Actually, DNA stands for deoxyribonucleic acid, or RNA stands for the ribonucleic acid. DNA and RNA, they both carry the genetic material, but there are quite different differences between them. DNA contains of the sugar called deoxyribose, while RNA contains a sugar called ribose. DNA is double-stranded molecule, while RNA is a single-strand molecule. And actually, when we talk about coronavirus, we're talking mostly about the RNA. And my colleague Antonio, when he talks about the testing using the PCR technology, using the genome, we will talk about a lot more about this. DNA is under the stable, is on the alkaline condition, while the RNA is not very stable. Now, next slide, please. Now, when we talk about the testing, we could divide them. Right now, here we are testing. We are dividing them by three different categories. The first one, and we are trying to, in each one of these categories, as we discuss them, we want to talk about the pros and the cons on each one of these testing. The first type of testing is the antigen testing, actually. And usually this is done by swabbing the nasal or the throat. And what you're detecting the protein exposed to the outer surface of the coronavirus. It works by looking at the protein rather than looking at the genetic material that the diagnostic or the PCR testing is doing it. Researchers, they don't expect this to be as accurate as that PCR technology, but it is possible that antigen can be used in screening patient and for infection. World Health Organization cautions the use of the rapid antigen test because of the science is new that the tests are unreliable or not proven. These tests are a lot cheaper, they're faster, and they can be done anywhere. A positive antigen results or test considered very accurate, but we get when we do these tests, you get a lot of false negative. What is false negative? False negative is that when the patient is carrying, somebody is carrying the virus, and the results comes negative. This type of test is not good to do it on the environment, surface, or air, as we're going to be talking about it in the next couple of slides. And the FDA has issued the first emergency use of the authorization for COVID-19 antigen test, and expect to have more tests to be approved and to streamline the process of approving and giving authorization for more antigen testing. Next slide, please. Now, the next slide, the next type of testing is actually the antibody testing. The antibody are produced when the immune system is fighting against a virus or a pathogen or illness. And by detecting the IgM antibodies, which develop alien infection, and the FGG antibodies, which shows later on, this is the principle behind the COVID-19 testing. Generally, this test could be done in few minutes with few drops of blood, but some researchers have used a more sophisticated testing instrument called ELISA, because which stands for enzyme linked immunoassay, which is more accurate as more expensive. Currently, we have about 12 antibody tests for coronavirus approved by FDA, but there are 200 more of them is under the consideration. Now, the drawback with the antibody testing is that usually the antibody testing, you only detect it when the person is infected and is in the late stages of the disease, basically. So if somebody who has just exposed to the coronavirus, it cannot be detected. This provides a very good information about the rate of infection in the community. Very little error in an individual results having less impact. But this also is very good for people who are recovering from COVID-19 and be eligible to donate their plasma. Some research has been done recently that some of the people that have been exposed has been recovered from the COVID-19, the plasma from them could be used for actually recovery and helping the recovery of people that they are struggling and dealing with the disease. And the next slide, please. And the next test we are going to talk about is actually the diagnostic or PCR based test. My colleague and Antonio is going to talk in more detail about this. But this is what we use as genetic material. They take the swab sample from some saliva or mucus, and this is the most reliable test that we have right now. Usually the samples are sent to the centralized laboratory, and you get the results back in a couple of days. But the actual test takes about 3 to 6 hours, and that depends on the automation. One of the drawbacks with this test is that when you take the samples from the throat or the mucus, it usually takes the virus, once it's passed on to the person, it takes some couple of days to get into their throat or to the nose. So somebody who just was infected, that is not the ideal test for it. And this is a very good test actually to do the environmental or surface or air test. We'll talk about it in the next couple of slides. One of the other things is they came up with a new Abbott Laboratories. They came up with a new PCR instrument that could be tested in 13 minutes. But one study has shown that you could miss up to 10% of the cases. As Paul mentioned earlier, in the slides, as the country starts to open up and we are going back to work, really the only way that we could open up the country and have a safe and be safe is to do more testing. Especially some of the industries that were impacted very badly with this COVID-19 testing. So we are talking about 4 different industries here. And the first one is the travel, tourism, and hospitality industries. As many of us, we are doing a webinar right now. Usually we'll be in the conferences, doing the conference, speaking on those. They have been affected very badly. The restaurants, a lot of hotels, the convention centers. So by having testing the air and swapping these areas, the kitchen, the hotel rooms, the convention centers, after disinfection, it really and having a fast turnaround time results and having the data posted and make it available to public, it really gives the customers and employees and helps the business. Another industry that was impacted hugely was the cruise industries, actually, with the coronavirus. The same thing with testing the air samples and the swab samples on the regular basis and making sure that after sanitation, everything is clean. That would give us another level of confidence and have people start calling and booking the cruise with vacations on the cruises. Next slide. Now, one of the industries that really hit bad was the food and beverage process plants, meat packers, meat processing plant. As we've seen and ourselves walking the supermarkets, we see freezers or refrigeration empty with any meat product, and you see that what the impact is COVID-19 had on food supply chain, actually. So by testing the workplace and the labor in agriculture or on dairy farms, on food processing plants, meat or poultry facilities, really it gives the employees a safe and avoiding disruption of the food supply chain in our country. And the next one is the assisted living facilities. And unfortunately, they were the one that took the biggest burden on the casualty of this COVID-19. And by the same thing here, by testing the surface and the air of these assisted living, after sanitization, it gives and posting these results on a regular basis on the website or on the door or anything. It really gives the family member, the caregivers, whoever works with them, an extra layer of the protection and being a protective response. Again, these are the 4 industries that we mentioned. It is not just limited to this. We have universities and public schools are opening up in August. That could be a perfect way and give a confidence to the parents and the students going back to the schools. The theme parks we're talking about. We talk about casinos are opening up. sports arenas. So there is a lot of different ways that this can be actually be tested and used. With that said, I'm going to pass it on to my friend and my colleague Antonius.
Thank you, Harry. Good afternoon to everyone, and thank you for joining us. I will speak a little bit about the technology that we use for the detection of SARS-CoV-2. This is the PCR, which stands for the polymerase chain reaction, a method that was developed in the 1980s. The PCR technique amplifies a genetic material like a DNA through an RNA through a series of heating and cooling cycles. During this amplification, different fluorescent DNA probes are attached on the targeted DNA, and as the DNA increases, the fluorescent signal also increases, and the PCR can actually monitor this signal, and this is how we understand if there is a virus in our sample. The COVID-19 test that we use is based on real-time reverse transcription polymerase chain reaction. This test detects 3 genes that are associated with SARS-CoV-2. The technique is very sensitive and it has a low error, less than 5%. The technique can be separated in 2 parts. In the first part, there is the synthesis of the complementary DNA, the cDNA, from the virus RNA. And in the second part, there is the amplification of this cDNA. Now, we can separate this procedure that we follow in 5 steps. In the first step, there is the extraction of the sample. And this is where we isolate the RNA that comes from the virus, and we deactivate it and put it in a solution. In the second step, we add the different reagents and primers and prepare our solution to be entered in the PCR. The next 3 steps happen inside the PCR, and the first step in the PCR is when we have the reverse transcription of the RNA and the cDNA is formed. In the next step, the cDNA is amplified, and finally the PCR detects any potential viruses in the viral RNA in the sample. In this slide, we have a floor plan of our mobile laboratory. As you can see, it's separated in three areas. Starting from left, we have the testing area. In the middle, we have the entrance area and where the scientists can enter and wear their appropriate PPE. And on the right, we have the viral hot area. In the viral hot area, the scientists can accept the samples and start the extraction process, in which they deactivated the samples in biosafety level 2 hoods and prepare the initial solutions. After they finish this extraction, they move on the left side of the mobile laboratory and in the PCR work hood, where they can add the different reagents and the primers and prepare the samples to enter in the PCR, which you can see in the green box. This whole procedure takes 3 to 4 hours, and with this procedure we can analyze up to 94 samples per run. When the PCR cycles end, the scientists upload the results in an automated lib system that was developed and customized by RJL Group, and the client can receive the results from an app. Now, there are many advantages that we chose to offer this test using a mobile lab. One of them is that we can use a technology, the PCR, that is considered the golden standard for the detection of SARS-CoV-2. We can also avoid any risks that come with shipping contaminated samples by having the mobile lab in any location that there is a hotspot, and provide results in only a few hours. And with that, I can leave the floor now to Paul.
Thank you, Antonius. I really appreciate it. The RJL Group has been using mobile laboratories for a number of years to solve difficult problems and also to provide results in the fastest possible and most accurate way. We understood over the years the advantages a mobile lab offers. It's probably the fastest way to generate accurate results. Best part is that a mobile laboratory and the way we approach the application of mobile laboratories is the fact that we can configure them to meet the specific requirements of the customer, the agency, the company that needs that kind of testing. Antonius mentioned that in 3 to 4 hours, we can have 94 test results generated. Well, that means if we have one shift of 8 hours operating within the mobile laboratory, we can generate twice as many. How about if we add 3 shifts? Now we triple the capacity. This is all based on a single PCR and a single automated prep system. We can add a second one or a third one. So as you can see, configuring the mobile laboratory to meet the precise needs a customer, an institution, an academic place, a hospital, a town, a city, is very, very important. Once we understand the requirements, it's very easy for us to reconfigure our mobile laboratory to meet those requirements and achieve the goals that are expected. It's always faster and better to bring the laboratory to where the testing needs to be done, as opposed to collecting the samples, shipping them, risking cross-contamination, taking much longer to do that. That's another reason why mobile laboratories offer the advantages that we are discussing. The data are almost immediately available. The way that we do it, we have a lot of experience in designing and implementing laboratory information management systems. We did the exact same thing for the COVID-19 analysis. We have our specifically designed and customizable LIMS system to meet that requirement. That allows for direct upload of data, actionable data immediately as soon as they become available. And obviously, when you have your laboratory at your fingertips, you can skip the normal supply chains, get things done a lot faster and a lot more accurately. So to us, the solution of performing COVID-19 testing using a mobile laboratory facility is the best solution that exists today. With that, what I can tell you is that just because we can perform the testing quickly, that doesn't mean we stopped our research and we stopped our capabilities. So I'll turn it back to Dennis, I'm sorry, Antonius, to tell us what's coming up down the line. Antonius?
Yes, thank you, Paul. So as we discussed previously, in addition to the analysis of nasal swabs that we mentioned with the PCR technique, we are working on offering more services in the near future. These services include the analysis of air samples that are collected using Coriolis air samplers or gelatin membrane filters. This media can be used to collect any virus that's in the air and then analyzed in the PCR using a similar technique as the one described before. Similarly, we can collect surface environmental samples using swabs, which can also analyze using the PCR. And finally, we're working on identifying the most efficient antibody test that we can provide for faster results in the mobile laboratory.
Thank you very much, gentlemen, for presenting today. Before we get to questions, here's a reminder that after we're finished with the questions, a survey is going to pop up. So a few seconds of your time will help us to offer better content in the future and let us know what you're doing. And we just like seeing that feedback. So give me 1 second to pull up our questions here. Here's a question. Tell me about what it's like for execution. How do we get a lab to the facility, and how's it configured? Tell me about the practicality. Maybe Paul, can you answer this question for us? Have you done it?
I certainly can. The best way to do this is to initiate a discussion with either myself or Antonios, and describe exactly the specific requirement that you have. We are very responsive. We will understand exactly what you're trying to achieve, and within a day we should have a proposal in your hands, telling you, based on our understanding, this would be the best customized or tailor-made solution to address the specific need. So in summary, reach out to either myself or Antonios, and we will get back in touch with you right away, scheduling an understanding of need and then responding from there.
Thank you. Here's everyone's contact information. I should have had this slide up. Here's another question we have. Does the mobile laboratory concept work in other places besides COVID-19? Like, where else do you have these?
Oh, absolutely. I can answer that again. We have been using the mobile laboratory applications for a long time. One of the major areas where a lot of our technology was developed is in support of Department of Energy activities, where we had to, in the past, we had to use technologies to identify very dangerous chemical substances in the air that could be affecting literally thousands of workers. And we outfitted our mobile laboratories with a different type of technology. This particular one is called proton transfer reaction mass spectrometry, where we were identifying all types of chemicals present in the air that could affect very, very severely the health of workers. Other applications were in the vaping industry. I know Antonios has done a lot of work in that area. We have even designed our laboratories to detect low impact explosives for non-nitrogenous based materials, things like TATP. So the applications of mobile laboratories are not new to us. They solve very difficult problems and they solve them quickly, very efficiently, and the technology really works.
Okay, thank you. Here's another one. My work site is a campus satellite layout. We have multiple locations, but in one general area. How would a mobile laboratory work for that? I'm paraphrasing here. Is that maybe for Paul as well?
Yeah, I think that would work very well because we have a couple of choices. If the multiple locations are very close to each other, then we can select a specific spot, park the mobile laboratory there, and begin testing by having people walk or doing a very short drive into the location of the laboratory. On the other hand, If we need to perform testing for 4 hours or 8 hours in a specific location and then move to the next location to which would not be too far away, it's easily done. All we have to do is physically start the laboratory, start the engine, park into the new location. Getting up and running is almost immediate, and we now can continue offering services. So it depends on the specific situation, but it's a very easy application, and that's why mobile laboratories work so well in this type of situations.
Okay. Here's another question. As I'm reading this question, I'm not sure if we'll be able to answer it. Someone asked, do you think it'll be long before we have a standard antibody test that always works? Maybe that's for Dr. Bazati, but I don't even know if we are even certified to answer that.
Antonios, maybe you have some idea, or Harry, you may have some idea.
I'm not aware of any that's really accurate as PCR testing. Don't forget, with the PCR testing, we are actually taking the genetic material, we amplify it, and we detected it. So not as far as I know, not as accurate as anything as accurate as PCR right now. Antonios?
That's correct, Harry. Currently, for antigen tests, the best way is the PCR, and there is no accurate antibody tests that we can definitely use for now. This is something that we're still researching.
Okay. One just came in. Is the mobile lab staffed with RJL Group staff for testing at a site? Paul?
The answer to that is yes.
Okay. Just one word, that works. I don't know if we can answer this one because of customization. A question is, how much is the cost of the mobile lab to come to your site for testing?
Well, again, it depends on the specific site, if it's a single employer with thousands of employees. But we have the ability to configure the laboratory to meet the requirements, and that configuration obviously can be paid either on a monthly basis or on a weekly basis or on a specific per day basis or even per sample basis. It all depends on the number of people that need to be tested, the frequency that the testing is necessary, and how much a laboratory like that would cost to configure. Obviously, the cost is different if we have one shift and one PCR versus 3 shifts and 3 PCRs. So it's a difficult question to answer, but it's easily answered once we have precisely the requirements put down on paper.
Okay.
I hope that answers the question.
Someone asked, the mobile lab is rented for use. Is that correct? Or is it a permanent lab on our premises?
Oh, it can be rented for use for a specific time period. And again, the ability to put mobile labs together the way we do it is to meet the precise requirements. So if you need it on a permanent basis, like we have a mobile lab for COVID19 testing, specifically for a very large customer and their very large workforce. But we can configure it any which way is necessary for the specific application.
Okay. I think we're good with questions. That's our last question. Again, thank you everyone for joining RJL Group today. I hope everyone out there is staying healthy. Thank you.
Thank you.
Thank you.
Thank you. Have a good day.